Hello, everyone. Welcome to today's webinar brought to you by Microbial Insights. This will be on prospects for PFAS bioremediation, inherent limitations, and reasons for optimism. We appreciate that you are taking the time to join us today, and it is my pleasure to introduce our esteemed speaker, Dr. Paul Hatzinger. Dr. Hatzinger is the Director of the Biotechnology Development and Applications Group at Optimism. He holds a PhD in environmental toxicology from Cornell University, where he studied the impacts of long-term aging on the biodegradation and bioavailability of pollutants. Dr. Hatzinger has more than 27 years of experience in contaminant biodegradation, bioremediation, and forensics. His research group has been instrumental in the development and field application of new in situ and ex situ remedial approaches for a variety of contaminants of concern to the DoD. His current research on PFAS includes studies of the co-metabolic and enzymatic biotransformation of precursors, efficacy of colloidal activated carbon barriers, and optimization of landfill leachate treatment processes. Paul has authored more than 100 peer-reviewed papers and book chapters and has served as the principal investigator on multiple research grants. Now, before I let Dr. Hatzinger take it away, I would like to remind you all, as always, this webinar is being recorded and it will be available on the Microbial website under the webinars section. If you go to the top menu, you'll see it there. After Dr. Hatzinger is done, there will be some time for questions. So feel free to type your questions as the presentation goes on. We'll try to read off as many as we can when that time comes so he can get you those answers. Join me in welcoming a true advocate for sustainable solutions, Dr. Paul Hatzinger.
Great, thank you, Marshall. And I would like to thank Microbial Insights and Dora for the invite today. I'd also like to mention the CertUp ESTCP program, who provided some funding for putting this webinar together through a tech transfer grant, and would encourage everyone to go to the CertUp ESTCP website to look at some of the different reports and projects. There are hundreds of projects going on looking at PFAS, including PFAS biotransformation. So let's get started here with a little bit of nomenclature. This scary diagram was put together by Chuck Newell, and I just kind of want to point out some of the different PFAS and what I'll really be speaking about today. And through most of this talk, I'm going to be talking about perfluorinated compounds. So these are the PFAS where each carbon is fully fluorinated. You have an N group, which can be often a sulfonate. So these are the sulfonic acids of different chain lengths, or this can be a carboxylate group in which you have the perfluorocarboxylic acids. Now, later on, as I get to the end of this, I'll be talking more about polyfluorinated PFAS, which are often called precursors. And these are compounds that have a fluorinated chain, but they also have some unfluorinated compounds. Here's an example of 6,2-FTS. And these behave very differently in terms of their biotransformation capability than some of the perfluoroalkyl compounds. So we'll sort of cover both of these. Now, to get started then, let's just review a little bit about why bacteria break down anthropogenic pollutants in the first place and what they do with these. And for bacteria metabolism, organisms, they need a carbon source, they need an electron donor, and they basically oxidize these to grow, so gain biomass, increasing number of cells, as well as produce energy and CO2. And many different compounds act as carbon sources, things like PAHs and alkanes and alkenes and BTECs. And very often the organisms that use these use oxygen as an electron acceptor and produce water. So when we have a BTEC site, this is the reason we try to add oxygen. We add electron acceptor and the organisms will use these carbon sources to grow and break down our contaminants of concern. Now for anaerobic degradation, there's also some electron acceptors that aren't oxygen, things like nitrate and sulfate. So there's organisms that can break down, again, things like BTECs, and toluene is a nice one in particular, because there's organisms that will use a whole range of electron acceptors to break down toluene. So that would be anaerobic degradation of a contaminant. The third thing that can happen is bacteria can actually use contaminants as the electron acceptor. So in this case, we provide the carbon source. So for things like PCE and TCE, perhaps we add emulsified oil. Organisms use that carbon source. and then they reduce these different contaminants of concern. Now, the other thing to remember is besides carbon and energy, organisms need inorganic nutrients, and they need a lot of nitrogen, they need a lot of phosphorus, and a little bit of sulfur as well as some micronutrients. So sometimes they can use contaminants, explosives, for instance, as sources of nitrogen. And then finally, there's this process called co-metabolism. And in this case, organisms break down contaminants, but gain nothing from the process. So they just have enzymes that are sort of fortuitously break these contaminants down. And some of this you'll see is certainly important for degradation of PFAS. So a little review there of microbial metabolism. Now, the question I'd like to pose, if we go back 40 years, we were really trying to understand the degradation of chlorinated compounds, chlorinated ethynes and ethanes. And it turns out, as we move ahead to the current day, it's widely accepted that these compounds are degradable as electron acceptors. And we know this organism was isolated called the Halicocoides. If we look at our metabolism, it uses acetate as a carbon source, hydrogen as an electron donor, oxidizes these to produce biomass, and then it reduces PCE all the way through to ethene as an electron acceptor. So it's quite interesting that, here's this organism, and there's a larger group of organisms that will take things like PCE through to DCE. We produce these compounds, and these organisms will be able to use those as electron acceptors for growth. And the other thing we understand now, we understand a lot of the genes that do this. They're often called dehalogenases. There's various things involved, but dehalogenases carry out this process of reducing and dechlorinating all of these different compounds. So we understand a lot about this process. So the question is, why does this occur for chlorinated compounds, and yet maybe we're not seeing it very widely for fluorinated compounds? I'm going to give you a couple of different explanations for that. The first being, a lot of these common dehalogenases that bacteria have likely have ancient origins, and that's because there are actually thousands of chlorinated organics present in nature. I recall going to an AGU meeting probably back in the 90s and going to a poster where someone was looking at natural production of chlorinated ethynes, and I was really surprised at the time. We're sort of looking at these man-made ethynes, but there's natural materials out there. There's actually thousands of chlorinated compounds, and natural enzymes create many of these compounds, halogenases, haloperoxidases, and they do this in many different species, even us. These are often used as a defense mechanism, and I'll show you, here's kind of an interesting compound which is known as halamin, and this is produced by red algae, and you can see it's a carbon backbone, but you've got a couple of chlorines here. You've also got bromines, so this is produced enzymatically by red algae, and we've actually looked at this compound, not my group, but over time as an anti-tumor compound. So some of these naturally produced halogenated compounds have very interesting potential applications. And basically, bacteria have evolved over the millennia to utilize these chlorinated as well as brominated and iodated organics. Okay, so as we start producing them, we start producing things like PCBs and PCE and all of these chlorinated solvents. Organisms had this metabolic framework. They had dehalogenases. They have this stuff in place, maybe with minor mutations, so that suddenly they're growing on compounds that we're producing, we think, for the first time, but that's not necessarily the case. It is certainly for some of them, but not for many of them. Unfortunately, these same dehalogenases generally do not defluorinate PFAS. All right, so let's look at fluorinated compounds in nature. Are there fluorinated compounds? As I said, there's thousands of chlorinated compounds. It turns out there's perhaps only a dozen fluorinated compounds that are naturally occurring. And this, if you think about it, this is pretty surprising. So fluorine is the 13th most abundant element on Earth, 13th. So it's more abundant than carbon, it's more abundant than chlorine. Just so you know, oxygen is number one, followed at a bit of a distance by silicon. But but it's a very common element. So there are some organisms that actually can add fluorine to molecules, and this is an example on the right. There's a fluorinase enzyme that does this in a variety of bacteria. So you're adding a fluorine and producing this 5-FDA. And interestingly, there's people who are now looking at these fluorinases to produce new drugs. The other part of fluorinated compounds, a lot of us, I'm sure on this call, think about AFFF and fire foams and stuff, but more than 40% of new drugs that are being produced are fluorinated. And this includes things like Lipitor and you know, chemists actually like to fluorinate these compounds because it can change the characteristics of a drug, and in particular, it makes it a little less metabolizable by us so that it can last longer in our system. It can change the PK, it can change the lipophilicity of the compound. So you see people adding fluorine and usually replacing a hydrogen. So there's an awful lot of fluorinated drugs that are going to currently are going to be in our wastewater treatment plants, some of which are going to be moving through into the environment as well. So just a little sidetrack there. But here's some of the compounds that are naturally fluorinated compounds. And one of the things you'll notice is they usually have just one fluorine, occasionally two. Now, one of these that is really interesting in nature is called fluoroacetate. And this is produced by plants, again, as a defense mechanism. And it's in tropical and subtropical areas. And it turns out in places like Brazil and Australia, a lot of the cattle losses, so when you have death in cattle, it's due to grazing on plants that have produced fluoroacetate, and it causes toxicity to the organisms. But there's not a lot of these fluorinated compounds, and they're usually just monofluorinated, usually at best difluorinated. Okay, so we know about dechlorinases. Are there enzymes naturally occurring that defluorinate compounds? And it turns out you can go back to the mid-1960s, and we understand that there are hydrolases and fluoroacetate dehalogenase that can defluorinate monofluorinated and difluorinated compounds, and they occur in a wide variety of different organisms. And it turns out, again, in the early 90s, there's a paper by Mary DeFlawn, there's a paper by Craig Criddle, but Mary was looking at this time, we didn't really care about PFAS in the mid-1990s. We did care about chlorofluorocarbons. And Mary, who I worked with at one time at Envirogen and went on to have a long career with Geosyntec, had published a paper showing that organisms, methanotrophs, can actually defluorinate some of these chlorofluorocarbons. And again, this is a co-metabolic process, so they gain nothing from that, but they can defluorinate these small compounds. So there is some precedent for defluorination, but the problem is, as we began producing these perfluorinated organics in the 1940s, so these compounds that had long carbon backbones that were fully fluorinated, bacteria really don't have an enzymatic framework to deal with these because, well, some fluorinated compounds occur in nature, and not a lot. They're usually monofluorinated or difluorinated at best. And these are very different animals from that. So they don't have that, enzymatic structure to deal with these. Now, the other thing that's really important when we start thinking about bioremediation is whether an organism can actually gain energy through degrading a contaminant. And if we look at the left here, we have our Dihalococoides. As I mentioned earlier, this takes hydrogen as an electron donor, takes PCE, we'll say, as an electron acceptor, and it can gain energy. So when you couple these processes, an oxidation reaction and a reduction reaction, there's a net gain of energy. So a negative delta G means that energy is available from the reaction. That's Gibbs-free energy. So let's make up an organism. We'll call it the Diphloracoccus. I hope it occurs somewhere in nature. We haven't found it yet. But this organism, can it take hydrogen just like the Dihalococoides does, and then take something like PFOA and defluorinate it? And will there be an energy gain? So that's an interesting question. So thermodynamically, can this occur? So we're going to have just another another review lesson, but just looking at enzymes, and enzymes have this function where essentially, if we have a set of reactants and a set of products, So as a reaction occurs, what an enzyme does is it lowers an activation energy. So that means as two, say, substrates are coming together to form a product, there's an amount of energy that goes in that gets you to an active state. And what an enzyme does is it provides an active site where it brings these substrates together, produces the product, and lowers this activation energy. So that's basically kinetic. So what an enzyme does is it speeds a reaction. What an enzyme doesn't do is change the thermodynamics of a reaction, and that's this delta G. So, whether this reaction occurs naturally without an enzyme or with an enzyme, the change in energy from reactants to products is the same. So enzymes don't change thermodynamics. That's sort of set in nature of the nature of these molecules. And when the products have less energy than the reactants, so we have a negative delta G, there's an energy yield there. So there's energy available, say, for an organism to grow. If this goes in the other direction, which it can, then energy has to be put into the reaction for it to occur. Okay, so now looking at this, some of you are probably familiar with this. Basically, again, we're looking at our electron donor, we're getting an oxidation reaction, we're looking at our acceptor, we're getting a reduction reaction. And if we look at these electron acceptors, there's this thing called the electron tower, and it looks at potentials of reactions. And without getting into too much detail, what we know from this tower is something like oxygen, which has a very high reduction potential, is a really good electron acceptor. So bacteria can gain a lot of energy by using oxygen as an electron acceptor. As we kind of run down this tower through nitrate and iron, and again, these are bacteria that would be using these as electron acceptors. These electron acceptors sort of get worse and worse till we get down to things like CO2 for methanogenesis, where organisms can still gain energy from reactions with things like methanogenesis, but the energy gain is not as great as it would be for aerobic respiration. And I'll just give you one sort of example of that. So here's just coupled reactions with benzene. So you're looking at the oxidation of benzene, producing, in this case, CO2 and water. So with oxygen as an electron acceptor. And what we see is that oxygen is an excellent electron acceptor, and these organisms can gain 3,200 kilojoules a mole of energy by growing on benzene, using oxygen as an electron acceptor. So you're going to produce a lot of biomass, and this also translates to very rapid degradation, typically, of your contaminants of concern. As we sort of run down with these reactions, again, still reduction of degradation of methane, or degradation of benzene. But as we get down to things like sulfate and methane, what we see is there's very, very little energy available. The organisms can still grow, but they're going to grow at very slow rates. So to get back to my original point then, let's think about chlorinated solvents. So how are chlorinated solvents on this redox tower as electron acceptors? Well, it turns out they're pretty darn good. So if you take PCE as an electron acceptor just through to TCE, that reduction potential is about positive 570. So it's not as good as oxygen, but it's still very, very good in the realm of iron reduction. And even DCE to vinyl chloride, again, it's pretty good electron acceptor. So you can see scenarios where organisms can couple hydrogen to the reduction of these materials, and they're going to gain energy. Now, one of the problems is, again, this is for some of these perfluorinated compounds, and this would be a reaction where you're taking that initial fluorine off of that backbone. These are very, very poor electron acceptors for that reaction. Not at all like TCE or PCE and DCE. And basically this is problematic because there are very few naturally occurring materials, including hydrogen, that an organism can oxidize and thermodynamically gain energy by this specific reaction. Now, I'll caveat that with there are some different, there's some different reduction potentials for bonds, and if you can get a fluorine off of this molecule, some of this will start to change. We'll talk about that a little bit with the perfluorinated or the polyfluorinated compounds as I move along a bit later. More good news. Do you want some more good news? Well, here's more good news. Fluoride is very toxic to bacteria. So when fluoride gets into bacterial cells, let's say they are defluorinating, you're going to need some type of transporter. Let's say a fluoride dehalogenase, or or you're going to need a fluoride transporter to actually remove that material. Now, the good news is that, again, because of a few of these naturally occurring compounds and because fluorine is a common element in the environment, there are quite a few organisms that have these naturally. So it's not something that is very, very rare, but an organism does have to have this if you're going to defluorinate PFAS. All right, so is there any hope for PFAS biodegradation and bioremediation? That's the question. So let's kind of move into some of the newer and sort of more interesting things that are going on in PFAS that may have some potential in the future. All right, so in summary, it's difficult for these organisms to grow. What about non-growth reactions? So we talked about co-metabolism. Is it possible that organisms may be able to co-metabolize some of these PFAS molecules, start to take fluorines off or break some of that backbone so that maybe the rest of those molecules become much more transformable? And the second thing is, can cells actually use energy? So when I started this, I talked about exergonic reactions, so reactions where organisms take reactants, they form products, and there's an energy yield. But can cells actually put energy into something to have a reaction occur? And is that a possibility for reducing PFAS? And the answer to that is yes. So here's a really nice example. When organisms fix nitrogen, now as mentioned, nitrogen is an element that bacteria need a lot of. There are environments where it becomes very limiting. So there's organisms that are nitrogen fixing bacteria. They have an enzyme called nitrogenase, and there's a series of enzymes. But what nitrogenase does is it takes this triple bonded nitrogen, so nitrogen gas, which again, these are strong bonds. This is a hard molecule to break, but it uses 16 moles of ATP, so it uses a boatload of energy to actually reduce this to a usable form. So things that you can put into proteins and DNA. Nitrogenase also operates at a very low potential, about negative 520 millivolts. So it works, way down on that sort of redox scale. So it just shows you that there is precedent for organisms to catalyze reactions and put a lot of energy in. Now, again, the caveat that I'll give you on this is these organisms need nitrogen, so it's worth them investing all the energy because they're getting the nitrogen they need to grow. One of the difficulties with the perfluorinated compounds is they're primarily composed of fluorine, which the organisms don't need, and carbon, which often there's quite a bit of around. So is there some great advantage to an organism to dump a lot of energy into degrading PFAS? I'll sort of leave you with that question. In many instances, that answer is probably not. Okay, so I'm going to step back a little bit here, and as we talk about PFAS biodegradation, I'm going to present a bit of a cautionary tale. And there was a nice comment by Carl Sagan, who was a professor at Cornell, billions and billions of stars, Carl Sagan. And he once said, extraordinary claims require extraordinary evidence. And that is still the case with PFAS, and particularly if we're talking about defluorination or detoxification of these perfluorinated compounds. And some of the questions you need to ask when these claims are made include, first of all, which compounds are researchers looking at? And I periodically have gotten excited about a paper, and it gets some press about defluorination, and it turns out people were looking at a monofluorinated compound, or they were looking at some of the polyfluorinated compounds, which I'll talk about a little bit. And you say, that's not quite as exciting as degrading some of the perfluorinated compounds. Second point is disappearance is not extraordinary evidence. An old colleague of mine, Rob Steffen, when he'd see a paper, he would always use the term disappearase for an enzyme, meaning it just seems to be something that disappears and we don't understand much about it. And I'll show you one example of this, but got to realize that PFAS, and particularly some of these perfluorinated compounds, they strongly sorb to media, they strongly sorb to bottles. And if the controls are not appropriately set up against whatever you're looking at in terms of biotic or abiotic transformation, you can be fooled by seeing something go away and it's not actually transformation at all. What it is enhanced absorption of some of these materials. Further, daughter products and fluoride are both hard to quantify. So a lot of times, you know, it's only university labs and really good ones that have the ability to quantify some of these unusual daughter products. There aren't standards for some of them, so you need people with a very high skill set to do that. And you're not seeing a lot of these materials by 1633 or any of the commercial laboratory methods. So when someone provides evidence of PFAS degradation, you know, you got to think about, were there appropriate controls? Did they extract whole bottles or were subsamples taken? Is there a mass balance? Are there pathways, and do those pathways make sense? And look for peer-reviewed publications. Okay, so moving on, let's talk about an interesting organism, the magic organism from Princeton, the Actinomycobium species strain A6. So Shan Wang and Peter Jaffe published a paper in 2019 that basically said, hey, we have this organism and it often occurs in a mixed culture. It's a bit hard to purify, but this thing defluorinates both PFOA and PFAS. And there was quite a rumble. Newspapers picked this up. It was a big news story. And then, as sometimes occurs, about 4 years later, a bunch of people came along and they said, hey, wait a minute. You know, we've looked at some of your data. I don't know why it took 4 years. They must have read the paper very, very carefully. But basically said, you know, we're not quite that happy with some of the controls. You know, we don't know that you don't have enhanced sorption on some of the iron minerals. We're not sure about some of the methods. So here's an example where we have a paper, and then we have some folks who are skeptical of the work. And I'm going to run through and show you some more current data from this organism and kind of talk a bit about what it does. So this is not an organism that in nature has just been waiting around to degrade PFAS for the millennia. It's actually an organism that it's an anaerobic autotroph, meaning that it uses CO2 as a carbon source. And in nature, it oxidizes ammonia or hydrogen and reduces a form of ferric iron called ferrihydrite. This is called the Fiamox process. It does well in acidic environments. It has been isolated. Again, Peter has isolated this organism, but it's hard to grow. Sometimes there's an art to growing these type of bacteria, which can be frustrating because then the work can sometimes be hard to reproduce. And basically, here's the reaction at the bottom. So all the things we talked about, again, there's a delta G yield here, so it can grow by, you know, reducing iron and oxidizing things like ammonia or hydrogen. Okay, so these days you can sequence a genome in a couple of hours and then sort of look at all the genes that these organisms have and say, oh, does it have any genes that would give it the ability potentially to degrade PFAS? It turns out it has some dehalogenases. It has a fluoroacetate dehalogenase. It has some other dehalogenase genes that we haven't seen before. It has some fluoride transporters. So you can take a look and you can say, wow, there's some interesting stuff here. And this is also important to try and get a further understanding of, you know, which genes might be involved in the defluorination, and therefore, you know, going further, are these things that you can clone? Can we look for them? Can we engineer better organisms with them? Why it has all of these dehalogenases, we don't know. But I will say about 10 years ago, we looked at this organism. I was working on some projects where we're looking at dibromoethane, which is an old fumigant and gas additive, and it debrominated that compound. So it does have the ability to basically debrominate things, and now it's been reported to be able to defluorinate some of these perfluorinated compounds. Looking at a little bit of data, so what we see here are, you know, PFOA down to PFBA. The blue is 0 days down to 100 days of incubation, and we see losses of some of these compounds, you know, much more so with PFOA and PFBA than, say, almost no degradation of PFPEA, a little bit of PFHXA, and we do see production of fluoride. So this organism is generating fluoride measured by multiple techniques. Again, I mentioned fluoride is hard to measure, but that's a very good sign because you're seeing the production of one of the end products that you would expect if you're seeing defluorination. Now, I mentioned pathways, and one of the original issues and the reason there was skepticism with this organism is that this compound was not originally detected. So the initial defluorination product, this HPFOA, was not apparently mentioned in the first paper. It now has been detected, and this indicates that the organism is likely doing an alpha defluorination, and then you can get some shortening of these molecules. And as you remove fluorines, if you can remove 1 or 2 fluorines from these backbones, these molecules can tend to start sort of breaking apart, or you can sort of get these cycles where you get smaller and smaller perfluorinated compounds. And here's basically a cycle that's been proposed. This was proposed by Larry Wackett, where you're sort of getting production of radicals and continued shortening of these materials. And I will also sort of give a shout out to Larry and some of his work. He's published several articles starting around 19 where he sort of looks at some of these different processes and thinks about thermodynamics and thinks about ways that PFAS can potentially be biotransformed. So I'd encourage you to go look at some of his papers. They're often open access and they're always an interesting read. Now, the other question is, well, where is A6 located? Is this something that, Peter just has in Princeton? Well, it turns out they're pretty commonly detected, often in acidic and iron-rich soils. So they're pretty widely found in nature, again, which is good and interesting if this organism truly defluorinates. And so the next thing that Peter's lab did is they said, well, can we biostimulate this organism in AFFF impacted soils? So again, you can go and you can do the type of thing that Microbial Insights does. You can see, is there acidomicrobium in a soil naturally occurring? If so, is there a way that we can stimulate it to degrade perfluorinated compounds? So in this study, they set up a couple of controls, one that just had some sediment with DI, one that was autoclaved that had PFOA or PFAS, and then 2 samples that had your sediments and they had PFOA or PFAS. And realize these are very high concentrations. We all understand this. You're looking for losses. This is not environmentally relevant at this point, but the important point is, can you stimulate an organism to defluorinate these compounds? So if we look to the right, here's PFOA. So in the treatment where you added all of the stuff that acidomicrobium needs to grow, there was a significant loss of PFOA. This did not occur in the autoclave control. So that's a good sign. With PFAS, the same thing occurred. Again, PFAS was added here along with all the nutrients for acidomicrobium. We see degradation. We did not see it in the autoclave controls. It wasn't added to these other materials, so these are sort of negative controls to make sure there's not PFOA or PFAS coming from somewhere else. And you see increases in A6 abundance. So this is all a pretty good sign because it suggests that, wow, you could potentially biostimulate these organisms in different environments to defluorinate some of these materials. Okay. So let's just summarize. So it's a hard organism to grow, but it is widespread in acidic, iron-rich environments, and it does have, I know, some tolerance at a slightly higher pH. It doesn't grow on PFOA or PFAS as far as people know. So it's a co-metabolic type process, but it does appear to defluorinate. You do see fluoride. It has some of the appropriate enzymes. And I'll say that the practical application is not yet clear, but I think this is a reason for optimism. And I think some of the early concerns that this organism, this was just an artifact, through additional papers and additional evidence, I think show now that, in fact, this organism is doing something, and it provides us with a little bit of sunlight in the perfluorinated world. And I'll also mention that Peter has received some additional funding from ESTCP to start looking at, not the basics in a beaker, but what you can start potentially doing at a larger scale with this organism. Okay, a couple of other enzymes and things that are interesting to look at. There are these enzymes that are called electron bifurcating enzymes. They're found in organisms in acetobacterium, in particular in wastewater treatment plants, and they're known to actually break down some highly fluorinated compounds, or at least defluorinate them. They work at very low potentials, like a nitrogenase. They participate in what's called caffeic acid breakdown. Now that is a coffee material, but it's not caffeine. They're different compounds. It has an active fluoride transporter. I've included a science paper here that talks a bit about this organism, but low potential enzymes, fluoride transporter, it has an ability to defluorinate some of these, relatively heavily fluorinated molecules. So again, an interesting compound, interesting enzyme. Now, the last thing are peroxidases and lactases. And these are enzymes, we know them because fungi in particular produce these enzymes in order to break down things like lignin. So very complex molecules, without these things, there'd be plant material piled up for miles on the planet. And because of these types of enzymes, they're extracellular. In some cases, you can start breaking down very complex materials. And there's groups, Kent Sorensen at Alonia, Shelley Mahendra, Jack Wang at University of Georgia, they all got funding where they're looking at different versions of these enzymes to break down perfluorinated and polyfluorinated compounds. And the other thing is we start moving forward and we see some of these enzymes, we're kind of in an age when perhaps you can do enzyme design. And again, there's companies out there, and Alonia is one that comes to mind, that are starting to think about designing or engineering enzymes. Where that goes, I don't think any of us know at this point. But we're sort of at a time when some of that can actually be done. So just looking a little bit at peroxidases, and I should mention, although I mentioned fungi, peroxidases are common. We have peroxidases. Horseradish peroxidase is a well-known enzyme from the horseradish plant that a lot of people study. So it's in plants. Bacteria have peroxidases. Mammals have peroxidases. So these are very, very widespread enzyme systems. And what they do is they take hydrogen peroxide and they create radicals. And in this case, this is an older paper by Colosi, and they were looking at PFOA and looking at horseradish peroxidase. And this compound, this 4-methylphenol, is what's called a mediator. And this actually forms a radical, which then potentially interacts with different compounds. And we see PFOA go down. We see the production of some fluoride. Again, these are good signs. We're seeing an end product. We're seeing loss of PFOA. And there's a whole bunch of potential daughter products detected. Now, a lot of these, I don't even know at the time sort of how they were doing this. A lot of us weren't thinking that much about PFAS in 2009, but they were seeing some very unusual compounds form, and they proposed a pathway, and a pathway where radicals sort of attack. And again, you kind of get this cleavage of molecules creating shorter and shorter compounds. And again, I think like anything, this still needs to be reproduced, but people are looking at peroxides and peroxidases as potential mechanisms to degrade some of these different compounds. Now, again, as you start to go through the literature, we're seeing an increasing number of reports of bacteria that are degrading compounds, in some cases perfluorinated compounds. And I'm just going to, again, put out my caution from slide 21. Go back to slide 21. And I'm not saying anything for any of these papers saying, oh, I don't think this is true, or I think this is true. I'm just saying it's good when you see these sort of papers to read them, be skeptical, think about some of the things that I talked about, energy potential, things that the organisms need to grow. And in some instances, when I look at these, I see, oh, organism A has come up in some of the samples where we're seeing losses of a compound. And what I always say to myself is the only reason that would occur is if that organism is gaining something significant from that perfluorinated compound. And what is that? So it has to be carbon, it has to be energy, it has to be perhaps sulfur or something like that. I'll talk about that a little bit, which it needs to grow. And in many cases, as I've already mentioned, there's not a lot of gain that organisms can get from some of these molecules. So I'm always skeptical if I see someone reporting some organism coming up and then claiming, oh, this organism increased in density, therefore it's degrading PFAS. You can't make that jump. I'm just going to tell you right now, and for the various reasons that I already mentioned. And I'll show you kind of one more thing here as we look at some of these different papers coming up. And a paper came out from Lisa Alvarez-Cohn's group at Berkeley, and they had looked apparently at some older papers attributing losses of PFAS and PFOA in a lactase system. And as they went back through and looked at this, they found it was really enhanced absorption. So they were able to reproduce this work, reproduce the loss of these perfluorinated compounds, but yet found that it wasn't biotransformation at all. What it was enhanced absorption of these in the system that had the enzyme compared to the control, which didn't have the enzyme. So there was an enhanced absorption process going on. And this is the important thing to think about, and what I mentioned earlier, where appropriate controls run, because these compounds are exceedingly difficult to work with in systems. Okay, so I'm just going to finish up talking a bit about some polyfluorinated compounds. Now, these are the compounds that don't have fully fluorinated carbons. Here's a nice example, 6,2-FTS, as I mentioned before. So it's a 6,2 means it's got 6 completely fluorinated carbons, 2 non-fluorinated compounds, carbons, and then a sulfonate group. So you have these guys around that don't have fluorines, and the behavior of these can be quite different than what I just discussed previously, which is the perfluorinated compounds. So they're present at most AFFF sites. So that's the other thing that's important to understand, is the compounds that you get by 1633. If you're a practitioner, you're running 1633 at your site, or EPA method 537, doesn't include a lot of these polyfluorinated compounds. Secondly, there's significant evidence of transformation from lots of different groups. There's a lot of work going on right now looking at polyfluorinated compound, and they're often called precursors because they're precursors to the perfluorinated compounds. And what I mean by that is what often happens to these compounds, they get released, they're in AFFF, the ECF, so the electrochemical fluorination versus the fluorotelomere-based AFFF. So there were 2 different ways that they made AFFFs, have different types of precursors, but often they have large functional groups. They're often tightly absorbed. You don't see them in your methods of analysis, and then all of a sudden bacteria come along, or sometimes abiotic processes, and they produce the things you do see. So this is one explanation for sort of the continued occurrence of some of these perfluorinated compounds, and the fact that sometimes they can be increasing as a plume moves along. I'll just sort of give you one example. This is from some of our work in a project that's collaborative between our group and Jen Guelfo and Andrew Jackson at Texas Tech, and this is some of the PhD work of Jessica LaFond. But essentially what we were doing is we were looking at 6,2-FTS, and it turns out you do see this compound at a lot of different sites as a precursor compound. It's one of the fluorotelomere precursors that's common, but it turns out it's probably what's called a secondary precursor. And there's some other compounds with very long backbones that can transform to this over time. And the other thing I wanted to mention that I'll show you in a minute, when you look at these molecules and a lot of the perfluorinated things that I talked about before that sort of end right here, there's not a lot there for organisms to use. But with some of these poly compounds, you've got nitrogen, you've got sulfur, you've got a lot of free carbon here. So if you can cleave this off, organisms can potentially use some of this for growth. Anyway, so what we were looking at is we were looking at some types of organisms that use propane and methane and other things that are traditional co-metabolic bacteria, methanotrophs, propanotrophs. They oxidize things like TCE and lots of other compounds. And we were trying to understand whether they would oxidize some of these polyfluorinated compounds. And what we found was that for some, but not all of them, they would take 6,2-FTS, they would add a hydroxyl group, which ultimately then became a ketone, and then that would further break down, through probably other enzymatic pathways so it gets shortened, and then you produce what do you produce? Unfortunately, what you're producing is terminal products are perfluorinated compounds. And if one starts to look in the literature at 6,2-FTS, what you're going to find is there's a variety of different organisms that will metabolize this. And I'm always hesitant to use the word biodegrade. I like biotransformation, and I think everyone should sort of think about that, because we're biotransforming this, in this case, to some products that are probably more resistant to degradation. Whereas a lot of time when we talk about biodegradation, we think of detoxification, going to CO2 or mineralization. But you see these products, but interestingly, for this compound and some others, you find that you consistently get some of the same products. So some of these pathways seem to generate daughters that are predictable, which I think is quite interesting. Now, the other thing that's interesting here is a paper came out, and there's been a couple of these. This has been reproduced with other organisms. This was out of Australia, and they were looking at an organism under sulfur-limiting conditions, and what it did is it used 6,2-FTS and an enzyme called alkane sulfonate monooxygenase, and it cleaved off this bond and used the sulfur. So it's actually doing kind of what I mentioned before. It's got a limiting nutrient. It cleaves off the sulfur and it uses it for growth. Unfortunately, it doesn't do anything with the rest of this, but again, it's sort of a means to gain a limiting nutrient, which I find very, very interesting. I will say in the work that we were doing, we had a lot of sulfate around and the organisms were doing this, so it was purely co-metabolic. It wasn't the organisms trying to gain a nutrient. And I'll just say there's a lot of these compounds out there, and that's something you need to realize. Some of these, even the acronyms are hard to say. So the PFAS, this is actually from a site, Jen Guelfo did this analysis, and it's looking at precursors extracted from an AFFF site, and there's a lot of these sulfonamides. FASA, this compound is a common one, which we see here among the sulfonamides, and then there's a whole bunch of others. And it turns out a lot of these are all precursors on their way, are being biotransformed or abiotically transformed through to FASA. So these are primary precursors, these are secondary precursors, and ultimately they're probably going on to form, again, other fully perfluorinated compounds as terminal products. So this was sort of a nice figure and just sort of putting some of this polyfluorinated materials or precursor transformation into context. When you have an AFFF site, you've sort of done fire training, you've got a lot of material often bound up toward the top of the soil, but you will get some leaching. But a lot of these precursors are going to sort of remain, and they're often, again, large compounds. They can be highly absorbed, but over time, biotically or abiotically, a lot of them will start to biotransform. And as they biotransform, you're going to start forming some of these perfluoroalkyl acids, perfluoral carboxylates. Some of these PFHXA is very common. In some cases, PFHXS, PFBA. You start seeing these things down gradient. So your polyfluorinateds are slowly transforming to some of your perfluorinated compounds. And as I mentioned, there's a lot of work going on in CERTUP. And the other thing I forgot to mention, there's actually a push to try and look at all of these pathways and get them documented. And there's a program called EnviPath. I believe it was originally developed basically to just look at pathways of contaminant transformation. And there's a big push within CERTUP, ESTCP, to start getting some of these pathways of precursor transformation into EnviPath. And this is being done at University of Zurich, and Chris Higgins, Colorado School of Mines, is also involved. So before long, we're going to see how common these pathways are, whether there's sort of pathways that are common and consistent for different compounds. And all of this is going to be important in our understanding of transformation of polyfluorinated compounds. So I'm going to finish up there with the precursors. There's a lot of biotransformation going on. Unfortunately, this is typically to terminal perfluoroalkyl acids. Now, for these PFAS, there are certainly some interesting organisms and enzymes, and I hope I've convinced you of that today. Lactases, peroxidases, whatever dehalogenase that Actinomicrobium is using. I think we're going to find other organisms that are doing this, but we also have to be cautious. And we have to be cautious because there's very little positive selection for defluorination. There's not some great thing that these organisms can often gain by defluorinating materials. They get a little bit of carbon, they get a little bit of fluoride, which they don't want, and you've got to put a lot of energy potentially into doing it. So keep that in mind. So these are often probably going to be energy consuming reactions initially, or they're going to be co-metabolic reactions, like in the cases of lactases and peroxidases. We also have to reach some pretty low levels. So while there are some limitations, I hope what this webinar has showed you also is that there's some reasons for optimism, and I hope that in 5 years we come back and there's 20 different organisms that are like Actinomicrobium that are defluorinating and transforming these materials, and we have a much better understanding than we do right now. And with that, if there are any questions, Marshall, I will entertain some questions, and like to thank everybody for spending, I guess, on either your breakfast on the West Coast or your lunch listening to the webinar. So thank you very much, everybody.
Thank you so much for that. And as you are well aware, it's a very popular topic, and so we have quite a few questions. But first, I wanted to thank you for providing all this necessary information on this as well. And I don't know, are you comfortable with sharing your slides with us so we could send those out to people?
100%. Yep.
Okay, great. So for those that are asking, you'll just email info@microbe.com, and we can provide you the slides once Dr. Hatzinger provides that with us, as well as the recording will be available on our website under the webinar section. So if you go to the top menu on the home page, there is a webinars section, and by the end of today, there will be a recording of this webinar. So to get off to the start, because acid microbiome is hard to grow in a lab, and the practical application is still being investigated, can you speak to the importance of transitioning from the lab into the field and maybe the need for cooperation from private stakeholders?
Yeah, absolutely. And I believe that, Actinomicrobium, again, it's an interesting observation at this point. It's unclear what the practical application is going to be. And again, and I'll also mention, this is what I'm representing is some of Peter Jaffe's work. He was kind enough to share that information, and I also find that he responds very quickly to emails. So I hope I don't overwhelm Peter's inbox, but feel free to shoot him an email if you have specific questions about that organism. But I understand that Peter is working with Ohlone at this point. I know that they're getting closer, they think, to isolating the enzyme that may actually be responsible for the defluorination, which may open up new windows. But in terms of practitioners getting out there and saying, okay, there's something I can do here to try and deal with a problem at my site, we're not close to being there yet. You can certainly look, and there are, it wouldn't surprise me if you found some of these types of organisms in lower pH aquifers, potentially aquifers that have iron. And I'm sure that, Peter and others will be looking, and Microbial Insights, I'm sure, can look for that organism for you. But what's the next step? So then it becomes, how do we stimulate, let's say, this organism there? How do we stimulate this organism to degrade PFAS? Now, how do we give it a selective advantage? Well, if you look at its metabolism, it would suggest that we have to provide things like ammonia or hydrogen. We have to have anoxic conditions, and we have to make sure that there's an available form of iron. So we'd be looking at maybe a biostimulation where we went out and did that. And Peter does, again, have some ESTCP money to start scaling. I think he's working with Ohlone to be thinking about this organism at the next scale. So those steps are happening. And certainly, if private companies, feel that there's a potential here, that investment becomes very, very important, particularly today. So, I think to answer that question, yes, scaling is important to the next step. Is there a bioaugmentation potential? This is a hard organism to grow. Is there a potential that you could grow it at large scale and have it survive? I can't answer a lot of those questions, but I think those are the next steps to think about. It may ultimately be a dead end. It may be something that in 10 years, we have sessions at Battelle talking about, some organism, this one or another one, just like we did with the halicocoides, how widely it occurs and how do you stimulate it, and how can we transform PFAS? I hope that's the case. I'm a natural skeptic with PFAS, but I do hope that's the case.
Excellent answer. And just to keep the ball rolling, since there's a bunch of questions coming in, if you could briefly describe what is the use of TFA in molecular biology?
Well, I'll give that one to you, Marshall. I think I'm not you would have to ask Dora, but I mean TFA is a very widespread compound in the atmosphere. I'll be honest with you. I am not a molecular biologist. I am an environmental microbiologist who does a lot of other work. So I will give someone else the ability to answer that. But I will say that trifluoroacetic acid is present at ppb concentrations in rainwater. It's purely an anthropogenic compound. There was a lot of debate at one time that there's naturally occurring trifluoroacetic acid from volcanoes and other things, but it turns out that it appears to be a natural compound. It's in the atmosphere. I know that some of the EPA folks are working on methodologies to detect it a little bit better. But in terms of the specific question, I'll let Microbial Insights send a written answer out to that so people can understand what TFA is used for.
That is definitely understandable. And like I mentioned, you can email info@microbe.com if you want that written answer more in depth on it. Everybody's saying very nice presentation, of course. And this next question is, are you optimistic with tropical soil environments potentially for discovery of novel species capable of transforming these compounds, or the energy need is a great limitation everywhere?
Yeah, I mean, I think again, I think the for some of these compounds, that the original ones I talked about, the perfluorinated compounds, I think those are ultimately going to be the most difficult. And what I tried to convey are some of the challenges compared to chlorinated compounds. So am I optimistic? Sure. And I think one of the things in one of Larry Wackett's papers that kind of stuck out to me is he said, these organisms are probably out there that can do some of these things, and they're going to be unicorns. Things like Acidomicrobium, they're not there because of perfluorinated compounds. They have some other metabolism, but fortuitously they can defluorinate one of these. Maybe they have to put energy in to do that initially, but once you can remove a fluorine or two, the chemistry of these molecules changes pretty dramatically. And so there can come a point where maybe there is an energy yield, if you can get through those initial steps. So that is a limitation. The other limitation, again, is that, when organisms need something like nitrogen, they're willing to put a lot of energy in. They don't need fluorine. They don't use it for anything. They need carbon, but it's not usually as much of a limitation as other things. So yeah, in terms of the incentive, the molecular incentive, the incentive to do that is not there for some of those perfluorinated compounds. But as I mentioned with some of the others, you've got nitrogen, you've got sulfur, you've got other things on some of those long end chains where organisms might be able to sort of grab those. The problem is you're often producing a perfluorinated compound as a daughter product. So am I optimistic? Yeah. And if you'd like to fund me to head to Brazil or Venezuela or Australia to collect samples and look for those organisms, I'd be more than happy to go.
Well, that is an excellent point as well. So this next one is, does strain A6 benefit from PFAS biotransformation/biodegradation?
Not as far as I know. And that would be a good question to ask, Peter Jaffe in terms of what's going on most recently, because it seems like, work with this organism and more people are starting to look at this organism now. As far as I know, this is purely co-metabolic at this point. So the organism isn't gaining carbon, it isn't gaining energy from the reaction. It's just a defluorination reaction that seems to be occurring fortuitously. If that's changed, I did send Peter my slides a week ago just to make sure that everything was still accurate. And as far as I know, it's not gaining anything from the process for PFOA or PFAS.
Gotcha. Well, hopefully he's not bombarded with questions as much as you might be alluding to him.
I could give out.
And we have reached the top of the hour, so I would like to express how thankful we are that you have presented this and answered these questions. And I know there are a few unanswered questions. So like I've expressed before, you can email info@microbe.com. So that way we can help answer those questions and make sure you are packed with this knowledge, as well as these slides that Dr. Hatzinger has provided. And if you have any other statements you want to make, Dr. Hatzinger, now would be the time. If not, we thank you as always for contributing to science.
Well, thanks, Marshall. And I appreciate the invite. I want to thank everybody for attending. My email is up on the screen, paul.hatzinger@aptim.com. If you have questions, something wasn't resolved, or if you think something I said was completely inaccurate, please feel free to email me. I will get back to you and we can have a discussion. But thank you everyone for attending. I know people are busy and I appreciate the fact that you're willing to jump on here for an hour. Thank you very much.
Thank you, and we'll see you all next month.